Hello everyone, welcome to today's session. Today we are going to see Class 12 Biology Chapter 5 Molecular Basis of Inheritance. In that we are going to see regulation of gene expression. So gene expression means the process by which the information which is present in a gene is used to produce a functional product. Usually it will be a protein or a polypeptide. Every gene in a cell does not remain active all the time. So a cell switches genes on or off depending upon its requirements, right? So this control of the gene activity is called as regulation of gene expression. And then gene regulation is also important during the development of multicellular organisms and also the development of an embryo into an adult. It involves the coordinated expression of many different sets of gene. So next we are going to see the regulating gene expression, the levels, right? So in eukaryotes, gene expression can be regulated at several stages. Let's see this stages. So here are some important levels of regulation. So here the first one is the transcriptional level. So what happens during this stage? See, regulation can occur during the formation of the primary RNA transcript from DNA. So from the DNA, yes, the primary transcript is formed. Then the cell can control whether the transcription of a particular gene takes place or not. And then next one is the processing level. So the primary transcript which came from the DNA, it undergoes processing to form mature mRNA. It forms mature mRNA and the regulation can occur during RNA processing. So it is including splicing also. We already know what is splicing. Splicing is nothing but introns are removed and exons are joined together. So what happens here? The primary transcript is forming mature mRNA. And next level is the transporter level. So the mature mRNA, it has to move from the nucleus to the cytoplasm. It has to move from the nucleus to the cytoplasm. And then the regulation can occur during the transport of mRNA. Yes, transport of mRNA. And the last stage is the translational level. So once this mRNA, it reaches the cytoplasm, it is translated by ribosomes to produce a polypeptide. So mRNA is now what translated to a polypeptide. So see this diagram, you can able to see. So the DNA, it is transcribed and it forms the primary transcript, right? So from there RNA processing takes place and the splicing also takes place, and then mature mRNA is formed and it is transported from nucleus to the cytoplasm. Last process is the translation where a protein or a polypeptide is formed. Let's see the operons and regulatory proteins. So in prokaryotes, the main control of the gene expression occurs at the transcriptional level. So the activity of the RNA polymerase, you can see here RNA polymerase, right? So at the promoter is regulated by proteins that influence its ability to recognize and also initiate the transcription process. And these regulatory protein, it can act in two ways. One is the positive regulation and another one is the negative regulation. So what happens in this positive regulation? Say activators help RNA polymerase and increase the transcription. What happens in negative regulation? Repressors inhibit transcription by preventing RNA polymerase from transcribing the genes. Then what is an operon then? See, operon is nothing but a group of structural genes. They are nothing but a group of structural genes, yeah, that are controlled together under a common regulatory system. They are controlled together under a common regulatory system. So here are some examples: Lac Operon, 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 His, and War Operons. These are some examples. So a operator is located adjacent to the promoter. So in many operons, the operator is the region where a repressor protein binds, and each operon has its own specific regulatory system. So let's see this diagram. So this is a typical operon. So this is the promoter, operator, gene 1, gene 2 and gene 3. So promoter is a site where RNA polymerase acts to initiate the transcription. and this is the operator. So the regulatory region can bind to a repressor. This is the regulatory region that can bind to a repressor. And these are the structural genes. So these genes are that code for proteins and is required for a particular function. So when the repressor binds to the operator, RNA polymerase is prevented from transcribing the structural gene. So if the repressor is binding means transcription is off. If the activator is acting means transcription increases, right? We'll see the next one. Anatomy of the Lac Operon. So the Lac Operon is an example of a regulated system in bacteria, right? So the Lac Operon was explained by Francois Jacob, and the biochemist Jacques Monod. So here the Lac, right? Lac refers to the lactose. So the Lac Operon, it contains one regulatory gene. It contains a one regulatory gene and three structural genes. So let's see this one is so I gene is a regulatory gene. I gene is the regulatory gene. So the I gene produces the repressor protein. What it produces? It codes the repressor protein. So the letter I does not mean it is an inducer, right? So it is derived from the word inhibitor because the gene produces a protein that inhibits transcription process. And next one is this Z gene. So the Z gene, it codes for the enzyme beta galactosidase. Yes, beta galactosidase. So this enzyme, it hydrolyzes the lactose. It hydrolyzes the lactose and it forms glucose and galactose. Next one is the Y gene. So the Y gene, it codes for permease. It codes for permease. So permease, it increases the permeability of the bacterial cell. It increases the permeability of the bacterial cell to beta galactosides and also help lactose to enter the cell. Next is the A gene. So this gene, it codes for trans-acetylase. It codes for trans-acetylase. So the products of all these three structural genes are involved in the metabolism of lactose. So let's see this diagrammatic part. So the Lac Operon, it can be represented as the I, right? This I, this is a regulatory gene. Yes, it gives the repressor. So the operon itself contains a promoter region. This is the operator, and this one ZYA. Yes, these genes are transcribed together as a single polycystonic mRNA. They are, yes, transcribed together as a single polycystonic mRNA. So remember these things, children, is very important for your board exam point of view. Switching the Lac Operon on and off. The Lac Operon is regulated depending on whether the lactose is available or not. So this lactose, it acts as an inducer. So the Lac Operon, it can exist in two important states. One is the off state and another one is the on state. So what happens in the off state? So when lactose is absent, the I gene is, it produces the repressor and the repressor binds to the operator and the operator is RNA polymerase is blocking it, and then what happens? Z, Y and A genes are not transcribed, and that is means is the Lac Operon is off. So what happens in the on state? The inducer is present. So when lactose or allolactose is present means it interacts with the repressor and the repressor becomes inactive, and the repressor cannot block the operator RNA polymerase. It can transcribe X, Z, Y and A. That means Lac Operon is on. So let's see this in detail. Let's see this off condition. So what happens in off condition? When lactose is absent, the I gene, yes, the I gene continuously produces the repressor protein. So the active repressor, it binds to the operator, that is the O region. You can see here. So when the repressor occupies the operator, it prevents RNA polymerase from transcribing the structural genes. These are the structural genes. So lactose is absent means active repressor produced by the I gene. Repressor binds to the operator and the RNA polymerase cannot transcribe and Y, Z and A genes remain off. Yes. So next let us see the on. So on, so when lactose is present means that's the condition on, right? So what happens? So when lactose is available, it enters, when lactose is available, it enters the bacterial cell with the help of permease. Yes, with the help of permease. So lactose or its isomer allolactose, it interacts with the repressor protein and makes the repressor inactive. So this is a repressor protein. It makes the repressor inactive. And now the repressor cannot bind to the operator and it blocks the transcription process. It's not possible. So the RNA polymerase can therefore it can transcribe the structural genes. So here lactose is present and then lactose or allolactose interacts with the repressor, yes, and the repressor becomes inactive and the operator becomes free and the RNA polymerase transcribes Z, Y and A and polycystonic mRNA is produced. So polycystonic mRNA, so beta galactosidase, permease and trans-acetylase are produced and lactose metabolism occurs finally. So we saw what is the off state we know very well and what is the on state also we know very well. So what we can say from this, if the repressor is active means transcription is off and the repressor is inactive means transcription is on. So let's have a quick revision of the topic. So gene regulation, so control of when and how much a gene is expressed. And the 4 levels in eukaryotes, that is the transcription, processing, transport and translation. Operon, a group of related structural genes regulator together. Operator is a regulatory region where the repressor binds. And then repressor is a protein that blocks the transcription process. I gene produces the repressor. I is nothing but the inhibitor. Z gene. So it produces beta galactosidase. So lactose, yes, it splits into glucose and galactose. Y gene produces permease and also it helps lactose to enter the cell. And the A gene produces trans-acetylase. And what is an inducer? So it can be a lactose or a isomer, that is a allolactose. So which inactivates the repressor and allows the transcription process. So we have come to the conclusion part. Yes, go through the video children. Yes, don't skip it. We'll see you in the next class. Thank you.